Hi guys, Miss Francis here to continue our discussion on DNA technology. Restriction analysis as a basic tool that's used in DNA technology, which was made possible by the discovery of restriction enzymes. Restriction enzymes are enzymes that cut DNA molecules at specific locations, and they were discovered in nature in bacteria. Bacteria will use restriction enzymes to cut foreign DNA. For example, the foreign DNA from a phage or phage, where a phage is a virus that infects the bacteria. Well, the bacteria doesn't want that virus, so it will use a restriction enzyme to cut it out. Scientists were able to isolate these restriction enzymes, and then they named the restriction enzyme based on the bacteria from which they were isolated. Restriction enzymes, just like other enzymes, are very specific, and they're going to cut the DNA sequence at a specific site or a specific point, known as a restriction site. So as I said, each restriction enzyme will cleave at a specific sequence of bases, where this unique sequence is known as a restriction site. And when these restriction enzymes cut the DNA, what they form are called sticky ends. They are pieces of overhanging DNA which then can bind to other complementary pieces of DNA, and then the various pieces can be fused using DNA ligase. So let me show you what I mean. This is called recombinant DNA. It is a cloning technique where restriction enzymes and DNA ligase can be used to make it. Where what it is, recombinant DNA, is DNA that's been spliced together from two different sources. So it's like recombining the DNA, hence the name recombinant DNA. So let's say I've got this original piece of DNA here, and I'm going to use a restriction enzyme to cut this piece of DNA at that restriction enzyme's restriction site, which is recognizing, or which has a recognition sequence. So the restriction enzyme cuts the DNA, and examples of restriction enzymes include EcoR1 or Hindi3. And what it forms are these sticky ends, where these sticky ends are just overhanging pieces of DNA. Now what I can do is take a DNA fragment from another source, cut it using the same restriction enzyme, and form the same sticky ends, which then can be glued together to form our recombinant DNA. So here's our recombinant DNA that contains our original piece of cut DNA, and then it also contains the DNA that was cut using the same restriction enzyme, but from another source of DNA. So why would I want to create this recombinant DNA? Well, let's say this other source that I'm speaking of has the DNA fragment that contains the gene that codes for insulin, for example. So recombinant plasmids are produced by splicing these restriction fragments from foreign DNA into plasmids. Remember, a plasmid is a circular piece of DNA that's found in bacteria. This plasmid then can be used as a cloning vector, which means that it can carry foreign DNA, it can carry a gene of interest into a cell and replicate there. So here's my plasmid, right? Here's my plasmid. It is a small circular piece of DNA. It can be used as a vector or a suitcase to carry foreign genes. Let's say, for example, the gene that codes for antibiotic resistance. Why are bacteria such as E. coli most commonly used as host cells for this gene cloning method? Well, because I can easily isolate the DNA. I can easily isolate that plasmid and reintroduce my now recombinant plasmid, which contains the plasmid plus my gene of interest, into their cells. Furthermore, E. coli or bacteria cultures grow rapidly and quickly, and as they're growing rapidly and quickly and dividing, each offspring also contains that recombinant plasmid. So this process of cloning a human gene into a bacterial plasmid can be divided into 5 steps. So the first step is to isolate our bacterial plasmid and to isolate that human DNA that contains the gene of interest. Then we need to insert that human DNA into the plasmid. And how do we do that? Well, the first step involves using restriction enzymes. I'm going to cut both the plasmid and my gene of interest using the same restriction enzyme, thus forming these sticky ends. And now using DNA ligase and enabling the hydrogen bonds to reform between the nitrogenous bases, I'm going to form a recombinant plasmid which contains the plasmid along with that piece of human DNA fragment that contains the gene of interest. Then I'm going to put the plasmid I'm going to allow the plasmid to do transformation. That involves calcium chloride and heat shock. Then I allow these plasmids, these bacteria cells to clone themselves. So how do I do that? First, I plate that cell onto a medium that contains agar, which has all of the nutrients that the E. coli or whatever bacteria I happen to use need in order to survive. And then what I'm going to do is I'm going to identify the clones that contain the recombinant plasmids, which therefore would also contain my gene of interest. So here are those 5 steps written out. So the first step, I'm going to isolate the vector, the plasmid, and the gene source DNA. By gene source DNA, I mean it's the fragment of DNA that contains the gene that codes for whatever it is we're interested in. Let's say insulin or the green fluorescent protein. So the source of the plasmid is usually E. coli, which is bacteria. And then the gene source DNA, the gene, can come from humans, might come from another organism, the insulin gene, for example, or the green or glow fluorescent protein gene. Now, the second step is to insert that gene of interest into the plasmid. So if I cut both the plasmid and that gene of interest, the human DNA, with the same restriction enzyme, I form compatible sticky ends, and then I can combine those 2 pieces using DNA ligase and complementary base pairing. Then I want to introduce that cloning vector or that plasmid into the cells. And this happens through the process of transformation, which involves calcium chloride and a heat shock. And we'll actually do this whole procedure in class. We're going to do a transformation lab. Then we're going to plate out the transformed bacteria and allow the bacteria to replicate on an agar plate. For example, an E. coli LB just stands for Loria broth. The Loria broth contains what the E. coli needs to replicate on a plate. And then I'm going to identify the cell clones that have the right gene. We're going to sort through the bacterial colonies that may or may not have the foreign DNA, because those that have the foreign DNA now contain our genes of interest.